Well, can everybody hear me? Is this on? Yep, that's great. Well, thanks, Nathan, for that very kind introduction. My name was mentioned a lot, but in actual fact, all of this is an amazing team effort. And it's great to see so many of my colleagues in the audience tonight, because without them, none of this would have been possible. And it isn't, one of the brilliant things about Edinburgh is not only the support you get from your colleagues, but the active engagement in all of these initiatives, which you don't often find in the university setting. So really, it would never, none of these would have been possible without all my colleagues, and it's wonderful to see you all here tonight. It's a great honor to give this lecture, especially following on from established colleagues like Richard Morris, who's in the audience, who gave a great Christmas lecture last year. And I just hope I can impart during this lecture what were the amazing time that we're living through and trying to understand not just the developmental disorders, but also disorders across the lifespan, because so many of the techniques that are used span, and it really relies on all of our colleagues, both those studying dementia or studying schizophrenia, as well as the autisms, that all of us working together are more likely to crack these codes. So the theme of the lecture, in the spirit of the holiday season, is really one of hope. And what I tried to highlight is the techniques and what we can glean from new techniques coming out, that actually I think there's real hope that we'll understand the brain, what goes wrong in these neurodevelopmental disorders, and eventually try to provide real, rational therapeutic interventions to address them. So the challenge that we all face in the neuroscience field can be summarized in this slide. So we know that neurological disorders span the lifespan. Just because something is a neurodevelopmental disorder, it's not that when the kids grow up, they all of a sudden don't have to live with this disorder. It's a lifelong disorder. And we also have the disorders of mental illness like schizophrenia and such, and the dementias. And these are big numbers with the worldwide incidence of these numbers. And the real challenge stems from this statement over here. These are all incurable, disabling, and progressive, and in some cases, fatal disorders. And currently the state of neurology is there might be a handful of rational therapeutic interventions. We have a big challenge on our hands to improve on that. And if you just compare this to the cancers, this is what's happened with cancer over the last couple of decades since 1990. If you look at the mortality rate of cancers, you can see that that's going down, that's declining. If you look at the number of cancer survivors, that's going up. And that's because in the field of cancer, they've been able to devise rational therapeutic interventions that are disease modifying, that target the source of the disease and can actually cure it in many cases. We can't do that yet. As I mentioned, there might be a handful of these types of medicines across all the neurologies. So the rest of my talk is going to focus on the neurodevelopmental disorders, the autisms, the intellectual disabilities, and the childhood epilepsies. And really, these are highly overlapping disorders. And whenever I put up a slide like this, where it gets into the genetics of it, whenever I give a public lecture, the parents in the audience say, those slides are great, but they really don't get across. They really don't capture what these kids go through and what the families go through. So in hearing from those families, and they nicely allowed me to use some videos from their kids, I just wanted in the first slide to really define what we're talking about. So these are 3 different disorders that we study in the lab: Rett syndrome, CDKL5 deficiency syndrome, and Fragile X syndrome. And these are 3 of the kids that, from the families that we deal with. So I'll start with Chelsea and Rett syndrome. So you can see that Chelsea had troubles with her breathing, and this is a classic seizure that Chelsea might experience during the day. Now this is Ava. Ava has CDKL5 deficiency. And Ava's experiencing a myoclonic event right now. It's a seizure. And the person's still alert. There's also a lot of self-harm that goes along with this, likely linked to the sensory hypersensitivity. You can see the bruising on her face there from this. And this is a video of Ken's. There's fragile. Listen to
Yes, you have to come down. No crying.
So what you can see in the case of Ken's and Fragile X syndrome, what I tried to highlight was the anxieties that go along with these disorders. Even trying to get Ken's to do something as simple as eat his dinner can turn into an hour-long process with these anxieties, which are very, very difficult to control. And I hope what you noticed as well with Ken's is he spent a lot of time covering his ears. Again, linking to the sensory hypersensitivities, and the deep stimulation is one way with Ken's that they found that helps to calm him down. And hopefully you can get him calm enough that he can actually leave the house and go for a cycle ride. So I hope what I've demonstrated from these videos is putting a face to these sorts of Venn diagrams. And the Patrick Wild Center, the Center for Brain Development and Repair in India, and now the Simons Initiative for the Developing Brain, we focus on the conditions that have that overlap. You can see that all of the kids I showed you will be on the autism spectrum. All of them have severe intellectual disabilities, and some of them have epilepsies. And there's a very good reason why we focus on this severe end of these spectrum disorders, is because in many cases, in fact in the majority of cases, if it's this severe, they have single gene causes. And we can identify the genes that are involved. For example, in Fragile X, it's the FMR1 gene. For CDKL5, it is called the CDKL5 gene. And for Rett syndrome, it's the MECP2 gene. And a very similar gene disorder is caused by the SYNGAP gene. And these are the ones I'm going to be talking to you about through the talk. But these single gene disorders can explain about 20 to 25% of all the autism spectrum disorders. And again, it will be the people at the severe end. We now know that there's about 873 risk genes. And this comes from the Simons Foundation Autism Research Initiative that have done big genetic screens to identify risk genes. And 80 of them would fall into the category that I'm talking about here that are single gene causes. So how do we go about trying to really untangle what's going on during brain development in these individuals with these very severe neurodevelopmental disorders? And I'll summarize it in what is often referred to as the virtuous circle, but I call it the potential of molecular medicine. And really this highlights not just what we do here at the University of Edinburgh, this highlights what researchers around the world try to do, and not just for neurological disorders, but for all medical conditions. In the case of the developmental disorders, you always start with the patient. And in order to try to understand what's going on, the first thing you have to do, as I've mentioned, is understand the genes. If they're genetic disorders, what are the genes that are underlying this and going wrong? we can then build models in the lab. And in many cases, the most common model that's used is the mouse. But for various reasons that I'll highlight in this talk, we've been able to branch out from the mouse. And that's one of the reasons I think it's a really exciting time to be studying neurological disorders. From these preclinical models, we try to understand the basic neurobiology that's going on and the disease pathophysiology to get a glimpse into what might be going wrong in the brain with the idea that we can one day correct it. Now, once you have the disease pathophysiology, you have to come up with theories of how you might be able to correct it. And those are incredibly critical to our understanding and pushing the field forward. And once we have those, we go back into the preclinical models. And we see whether we can improve brain development in these models. And this process is an iterative process and takes a very long time. And what you hope is through understanding the disease pathophysiology, we can eventually come up with pharmaceutical development or intervention strategies that will actually eventually go on to clinical trials. And this is where we are currently. The state of affairs for something like Fragile X is there's currently about 12 clinical trials going on around the world. And I'll briefly touch on those at the end. But I'm a fundamental researcher. I don't run clinical trials. I leave that to my clinical colleagues. What I want to do, and what I'm going to try to cover in the rest of this talk, is how do we understand what's going wrong in the brain? And for most of these disorders, or for many of them, we don't see any outward differences in the gross structure of the brain. In some cases, the brain can be a bit bigger, in some it's a bit smaller, but we largely, just from looking at the brain, can't tell what's going on. So we have to delve deeper. And we can do this with a range of techniques. We can look at the circuit in humans using functional imaging or diffusion tensor imaging, which shows all of the amazing connectivity of the different regions of the brain talking to one another in the region and how they're interconnected. From here, we can delve even deeper using very high-powered microscopes and start looking at the individual cells and neurons. And there's 100 billion of these in a human brain. This happens to be taken from a photograph that Manny Jackson took of a neuron in the cerebellum. And you can see it in all of its beauty. So you can see this big tree-like structure, which is referred to as the dendrites. And these are essentially the receivers for these neurons. This is where all of the inputs, how all the other neurons in the brain can talk to them, can talk to this neuron. It then has to integrate all of the information coming in and pass it on, and it does that through the beginning, which is cut off here, but a process known as the axon. And the axon then sends out and makes connections with thousands of other cells. But in order to understand these disorders, we have to delve even deeper. And we have to start understanding what's going on in this receiving bit, the dendrites of the neuron. So we can zoom in, and we have amazing techniques. This is super resolution microscopy that's been done. But originally, this came from Lhasa Wijatunga in my lab. And using this, we can get a very high magnification of these dendrites. And you notice that they have these protrusions coming up, these hair-like protrusions. These are called dendritic spines. And each one of these spines is receiving a synapse from another neuron. And that neuron that I showed you in the cerebellum, the Purkinje neuron, that receives tens of thousands of inputs. Other neurons, which I'll show you examples of later, receive about 1,000 inputs, but somehow they have to integrate all of this and decide what information they're going to pass on. And we can even zoom in now on the individual synapses, and we can look at the molecules within them. So this picture is taken from a mouse brain, from a mouse that Seth Grant and his colleagues made. And each one of the greens represents a molecule in one of those spines. Similarly, each one of the red dots is a molecule in a different set of spines. And the blue dots represent a presynaptic axon coming in and talking to them. And to give you some idea about the synaptic complexity in the brain, each one of these spines in terms of their size is about 1/2000th of a millimeter. So they can be packed in pretty densely. And there's over 100 trillion of them in the human brain. And if we take that number and we scale up that size to the size of a penny to give you some idea of the magnitude we're dealing with, if we lined up 100 trillion pennies side by side in the equivalent number of spines in the human brain, it would go to the sun and back 13.5 times. That's how many synapses are in your brain. So the classic way that we go about trying to understand this is what I would call a bottom-up approach. We start with the molecules that are located in the brain, and we understand how they function at a synapse. We then try to understand how any changes in the molecules or the synapses are integrated at the level of the cell. We then look at how the cell integrates within a circuit. And finally, ultimately, we look at behavior. And this is a classic sort of bottom-up approach for understanding the brain. But there's also a new move afoot, which is that we can use top-down approaches. So we start with the behavior in our animal models, and we start trying to understand the circuit that underlies that. And eventually we can move down to what's going on in the cell and to the molecules. And indeed, this top-down approach is what we've invested heavily in the Simons Initiative for the Developing Brain. And we do both approaches. We do the bottom-up, we do the top-down, and we're hoping that they join in the middle and that each one will inform each other to get to this global understanding. So we do that from the point of fundamental research, but it's also, the bottom-up and top-down approach are also useful to think about in terms of how therapeutic interventions might work. So the classic bottom-up approach to therapeutic interventions would be your gene therapies or pharmacological interventions that really target the core biochemical pathways that we believe are going wrong in these disorders, either replacing the gene or trying to rescue some of the pathways downstream of that gene. But we can also start thinking about some top-down approaches. And rather than targeting the core biochemical pathways, if we can understand the circuit, what's going on at the circuit level well enough, then we can immediately go in with pharmacological tools and try to tweak the circuit so that we get a better output and try to get improvement in the behaviors that I've shown you, be it the anxiety behaviors, be it the epilepsies, whatever, these outward expression of the disorders, we can start trying to make a difference at the circuit level. So there's various ways of doing the top-down and bottom-up approaches, and we're doing both of them here in Edinburgh. But the real question, I really want this lecture to be more about hope, right? It's the festive season. It's a season of hope. So that's what I'm going to try to get across to you in the next bit of this lecture, is why I'm hopeful that we can really start to understand what's going on in the brain in these disorders. So essentially, it's why now? Why is this a good time to be doing it? And why do I find this an amazingly exciting time to be a researcher in this field? Well, ultimately, what I've told you is we need to understand brain function and dysfunction in order to develop rational therapeutic interventions and eventually, hopefully, either prevent the emergence of these disorders or be able to reverse them once they've started. And I think there's been three major advances that have gone on, or three fields that have had major advances that are impacting the way we understand brain function. One is the genetics. I'm going to show you a slide in a minute which illustrates how far we've come in being able not only to understand the human genome, but in the ability to manipulate genomes, and how that's had a huge impact on this research. We've been able to develop new models. Obviously, the best model for a human is a human, and we now have models which Siddharthan is using in his lab and developing in his lab, where we can actually study human neurons in a dish. And that's a huge advance for our ability to translate through what we learned from our animal models through to the clinic. And finally, imaging. You've got a glimpse of what the types of microscopes we now have and our ability to image the brain and in some cases inaction, which we've never had before. And all three of these have had a major impact and will have a major impact in the coming decades. So I'll start with the genetics. In order to understand how far we've come in understanding the genetics, we really have to just go back about 60 years. And I'm highlighting this along a timeline of genetic discovery. If we start with what was about 65 years ago, the identification of the structure of DNA from James Watson and Crick. And this was a fundamental sea change in how we understood heritability. It was a bit of a quiet time after Watson and Crick, but in about 1983, that was when we first started to be able to associate particular changes in the DNA sequence with human disease. And the first one was Huntington's disease in 1983 was mapped to the long arm of chromosome 4. And in 1989, the cystic fibrosis gene was identified, and it was the first gene to be identified as a cause of human disease. What this started was a revolution that made us realize that if we understand the genetics, we can understand many human conditions. And it initiated in 1990, the Human Genome Project was initiated to sequence the entire human genome. And it took 13 years to do that, and over $3 billion of US funding. And this was a worldwide project, so add on top of that the money that was spent at the Sanger Center here in the UK, which was another $500 million approximately. It was a huge endeavor. And essentially what we found out was that the human genome has about 3 billion bases. So it actually cost well over about $1 a base to sequence this. It took 13 years. Now slowly, what we learned from this and new techniques being developed, we started to be able to associate other genes with diseases, but it was a slow process. So all over this time, people were coming up with new genes associated with human conditions, but it was slow. So in 2004, one new gene was added in that entire year. They identified one more gene for a human condition. By 2005, 5 new genes were identified for human conditions. And in 2006, 8 new genes were discovered. So this was a rather slow process. We were getting there, but we needed to speed things up. And a major change came 5 years ago with next generation sequencing. And all of a sudden, we could sequence a human genome in a day. What took 13 years initially, now, only a decade later, was taking a single day. And as a result, we were able to, we now know over 6,000 genes, and hundreds of thousands of genomes have been sequenced. Right? So it took 14 years to go from sequencing one human genome to being able to sequence 6,000 or hundreds of thousands of genomes. And it's estimated that another genome sequence comes off the sequencers every 15 seconds. So it was an amazing change in only 14 years. So no one, I don't think, would have ever believed. I certainly didn't back when they were starting the human genome, and I remember it well when this amount of money was committed to it. No one would have thought that within 14 years we'd be able to do what we're doing now. So what's going to happen in the next 14 years? I don't think any of us can say. I don't think we imagined it back then. The capabilities are really endless, and I'm really excited about this. So, and one of the things that we now know, which is not mentioned on this slide, is now we have the ability to specifically edit genomes. And I'll come back to that a little bit later. So, what does this have to do with autism and all the neurodevelopmental disorders? Well, it was around 2010 when I started getting into studying these disorders, and it really came from papers like this one. This was one of the first papers that came out from a British group in London, but it was a multinational consortium. And it started to identify the genes that are involved in people with severe autism. And I was in my lab working on mouse models with single genes where we can remove a single gene to understand how the brain was developing. And at that point, we had about 5 or 6 different models in the lab. And literally overnight, every single one of those models became a model for autism and intellectual disability and epilepsies. We were using them because we knew they were involved in brain development, and we wanted to understand the basic mechanisms by which the brain was developing. And we always used to write at the end of our grants, if we can understand this, one day it will help us, it will tell us a lot about neurodevelopmental disorders. And overnight with next generation sequencing, that became true, and it went at the front of our grants. And not surprisingly and correctly, we started to get more grants. And what this also told us was that the nature of the changes in the DNA that lead to this can be very small. So here's an example of a single base pair change that leads, this is the protein sequence, but a base pair change, 1 in 3 billion base pairs changed in a gene led to the change of a single amino acid, and this led to, in this case, schizophrenia. So this is a high-risk gene for schizophrenia, but exactly the same premise occurs in the autism. And these are de novo mutations. All of us, there's about 50 new changes in the DNA that did not come from your parents for everybody that's born. On average, there's 50 new, what we call mutations, but 50 changes in the DNA. And if it happens to be in one of the genes, one of those high-risk genes, then the likely outcome will be autism. So it seems a bit daunting, right? It seems a bit daunting. We have 800, or about 900, 873, 900 high-risk genes for autism, or risk genes for autism, and about 80 that we know if there's a change in that gene that affects the protein function, the high likelihood is that you'll have autism. That seems like a lot. So do we need a bespoke treatment for each one of these different genetic causes? Are we going to have to understand each one individually? And again, looking at the genetics and all of the biology that's around that we already knew about these genes, we actually don't think that's the case. So if you just cluster these genes based on what they do, you can see that many, many fewer clusters come out. In other words, we can stratify the autisms based on the type of gene that's involved and the known function of that gene. So in many cases, the biggest cluster is synaptic function. All of these genes were pulled out of the genetic studies of cohorts of people with autism, and all of these are synaptic function. And this is where Fragile X, the SYNGAP, we believe the CDKL5 would fall into these groups. Another big group is the chromatin remodeling, and the chromatin is essentially the structure of the DNA. And these genes regulate how the DNA is expressed, eventually into the protein. So they regulate gene expression. And this is where MECP2 would fall into this one, or the gene that underlies Rett syndrome. Okay? So we think that because we can cluster these, and they're doing similar functions either at the synapse or regulating the chromatin expression, the genetic expression, that maybe developing something for SYNGAP will also be an effective therapeutic for these other disorders. But as I mentioned, currently we're in a state where we don't have any effective therapies for autism. So that's the genetics. And I hope you get a feeling that our understanding of the genetics is setting so good that the next decade is going to be really exciting. The next thing I'm going to show you is about imaging. Now, when I was doing my PhD, the only way to image the brain was to cut it up, right? And even then, we had the microscope, the resolution of our microscopes was so poor that we really couldn't visualize those spines that I was talking about. And that's all changed. And I hope in the next bit of the talk, you'll see how exciting it is for us to understand brain function through imaging. And in this case, I'm talking about imaging of animal models. So what I'm going to show you is our endeavors in the lab to really understand the sensory hypersensitivities associated with Fragile X syndrome. It's one of the main things we study. And this is Ken's again with his parents, Gus and Reem, and this is Ken's in a much happier mood. So what I'm going to show you is our attempts at the bottom-up approach. And really I'm going to take you from the synapse through to the cell and eventually onto the circuit, an understanding of the circuit. And because I'm focusing on the sensory hypersensitivities, everything I'm going to show you comes from the somatosensory cortex of the mouse. So this is the region of the mouse that gets input from the whiskers, right, which is the main sensory modality that's used by mice. And here is a cell in the cortex from a mouse. And this is in a part of the cortex. These cells get the first information coming from the whiskers. And we had what we thought was a very simple question, which is to say, how do these neurons differ in a mouse model of Fragile X compared to a typical, or what we call a wild type mouse? And really, what we were asking is, how do the dendrites receive information and integrate it? And how does this cell integrate that information and decide what information to pass on? And essentially, what we want to do is listen to that cell and see what it's telling us. So the way we do that is this is a long glass electrode that's been filled with a fluorescent dye. And to give you some idea of what's going on here, this cell body is about 10 microns apart, or 10 microns across, or 1/100th of a millimeter. So this is a very fine glass electrode that we can patch onto the cell, and now the dye flows out and we can see all of the beautiful arborization of this cell. And this is an electrode, so we can also listen to what that cell is telling us. And this is a classic physiological recording that we get, and each one of these downward deflections is one of the synapses firing, or a couple of the synapses firing onto this cell. But what we've never been able to do is to actually associate each one of these events with a particular dendritic spine. And this is what we want to be able to do if we're going to make any generalizations about how does spine function or synaptic function change in these models. So to do that, we relied on the advent of the two-photon microscope, which is a very high-powered laser-based microscope, where we can fill these cells, and now using a laser and some clever chemistry, we can shine the laser at a particular point anywhere that we want and only have the neurotransmitter that's used by these cells, glutamate, only have glutamate activated at these points. And when you do that, here's the physiological response you do when you do this series of stimulations. And you can clearly see that you get a beautiful response when it's just out by the tip of the spine, which is where the synapse happens. And now we can say, yes, this particular spine with that particular structure gives us this size of response, right? And we get a beautiful structure-function relationship. So now we can associate with each spine an individual physiological event and ask, how does that differ across this neuron in a Fragile X model? And will it give us clues to those sensory hypersensitivities? And this is the data, and this was all done by Sam Booker in the lab. And you can see that when you uncage, or when you do the stimulation in a Fragile X model, you get a much bigger response on average at these spines. So perhaps the synaptic dysfunction that's going on is at least one of the components that's leading to these hypersensitivities. The cell is getting too much information, essentially, and passing on too much information. But we really needed to know whether that was the case. So what we need to do is understand how all of the different synapses going on here, how all the different inputs from around the brain are integrated in this cell and passing on the information. And to do that, you have to understand one more thing about the dendrite. It just looks in these pictures like it's a tube. but this has to conduct electrical signals. And the way it conducts electrical signals is through ion channels that are spread throughout the membrane. Okay, and this is just a schematic of that from Matt Nolan. Each one of these dots represents a different ion channel. And these ion channels not only pass the electrical signal along the dendrite down to the cell body to decide how it's going to fire, but they also regulate the overexcitability of the cell. Okay, how excited it's going to be. And one of the key groups of ion channels that do that, I can give an analogy to this hose with all these holes in it. And these are what we call leak currents. Okay? So what goes on, you can just imagine that you turn on a hose, and if the hose has no holes in it, that the amount of pressure you put determines what comes out the other end. And if the analogy for the neuron, how many action potentials it's going to fire and pass on to the next group of cells, what that information is. Now obviously, if you have a bunch of leaks in your hose, less water will flow out the other end. In the neurons, if you have fewer of these leak currents, then it won't pass on as much information to the next cell. So we really wanted to know what happens to these ion channels that are regulating the excitability of the neuron. And if we look at that by blocking these leak currents with chemicals, in a wild-type animal cell, you get a big change in the amount of current that comes through. You get far less in the FMR1 mutant, indicating that the FMR1 model has fewer of these leak currents, so it's passing more electricity down and passing more of a signal down. So what I've shown you is that we have the synapses themselves are overfunctioning, passing too much current. The dendrite is unable to relieve that electrical excitability and is becoming overexcited. But what we really want to know is what is the output of the neuron? Is the output of that neuron, the action potentials that fired too much, which would help us explain the sensory hyperexcitability? And indeed it is. So Alex Domansky in the lab did this very simple experiment. I call it simple. Anyway, this very simple experiment where he patched the neurons and listened to the output of them, but to various stimulations. And this is mimicking, it's in a slice, brain slice in a dish, but it's mimicking what would happen if you stimulated the whiskers. Okay? And you can see even these very low stimulation frequencies, which in a wild type animal, in a typical animal, would not induce any action potentials in these cells, is eliciting a huge number of action potentials in the FMR1 mutant. So indeed, what we've isolated is what we think is part of the core physiological deficits that are leading to these sensory hypersensitivities, which would never have been possible 10 years ago, but has dramatically increased our understanding of what's going on in the brain when you lose the FMR1 gene. So I've talked to you about this bit here. Now I'm going to talk to you and a little bit about the circuit, because the output of that neuron is essentially its functioning in the circuit. But really, we're listening to a single neuron, and we want to be able to see what's going on in the whole brain, or at least in a small region of the brain. And using these 2-photon microscopes, people have now been able to start watching whole ensembles of cells in the brain in an awake animal while it's doing a task. Now this is amazing. Again, when I was sitting in Colin Blakemore's lab in Oxford, cutting section after section, trying to understand dendritic spines, I never believed that within 2 decades we'd be able to watch whole neural networks in action. And I'm just going to show you that in the next slide. So here's a mouse. You can see it's wide awake, and it's doing a visual task. So you'll see a bright light go on, and that's when we're showing this animal on a screen different visual stimulations. And then over here what you'll see is this is 2-photon imaging of the cortex using specific dyes that we can now use. And you'll see the neurons flashing, and this is the visual cortex in action while this animal is seeing this task. So there the stimulus is going. It's also on a ball, so it can run, and we can look at the integration of the visual stimulation with its motor task. And you can see these neurons firing, and we can start asking amazing questions about how this circuit works and how it goes wrong in an animal that lacks FMR1. And I'll just make one brief point here. We're doing this is all work done by Thanai Katsanavaki in the lab. And she's been working on another single gene cause for autism and intellectual disability, which is called SYNGAP. And for various reasons, we had reason to believe that SYNGAP and FMR1 converged on the same biochemical pathways, but we didn't know about this circuit. And much to our surprise, when we looked at this, rather than being hyperexcitable, which is what we see in the FMR1s, they were hypoexcitable. So the circuits in the SYNGAPs are hypoexcitable, and we're trying to tease apart how you go from a similar looking biochemistry to a very different looking circuit. But none of these questions could have been asked even 10 years ago. But there's one main problem with even this beautiful two-photon imaging, and that is that we're absolutely restricted with this two-photon imaging to a brain area that we're interested in. With a visual stimulation, it makes sense to look at the visual cortex. But with more complex behaviors, we don't know necessarily, we have some ideas, but we don't always know exactly what brain areas to look at. And just like when, and again, coming back to the complexity of the connectivity, the brain doesn't always use one pathway or one group of connections to solve a task, let alone is they're using the same brain connectivity in an animal model of autism. So we need a way that we can unbiased look at the circuits that are being activated in our animals. And just like when I walk to work in the morning, sometimes I want the scenic route and I walk through the meadows. Other times I want a coffee from Starbucks or another coffee shop and I walk down through Newington. I still get to work even though I've used a different pathway. And that's essentially the question we're trying to answer in these animal models. What are the pathways that they're using, and are they similar or different? And what is the case when we see alterations in their behavior? So there's the major problem. There were two major problems that had to be overcome to come up with an unbiased way of looking at brain circuits during different behaviors and in different animal models. One was that the brain is opaque, and this is a mouse brain, and you can see that it's opaque. You can't see through it. And that's because of a group of molecules in the brain called lipids, right? They diffract light. So if you try to shine light through the brain, you can't because the lipids diffract it all. So the first thing we had to do in order to get access to being able to see them was we need to make these permissible to imaging. And some very clever people developed a technique with the great name of clarity, and they were able to get rid of the lipids in the brain, leaving all the proteins and the cell locations intact. And that's essentially what you see right here. So they developed a technique where you could clear, and now this brain is accessible to light-based microscopies. There's still a problem of throughput. How do you actually image the brain in a very fast way so we can take animals at different points from their behaviors and really understand the temporal sequence of how the different brain regions get activated. And that was solved by another group of very clever people who developed something called light sheet microscopy. Similar to two-photon microscopy, it gives you depth, a certain degree of depth within the tissue, and now the tissue is cleared. But it creates a light sheet that you can rapidly scan through a whole brain. And if we combine these with techniques that allow us to label up, genetic techniques that allow us to label up neurons that have been active, we can scan through in a very high throughput way. It takes about minutes to hours to scan through a big piece of tissue. We can now have a high throughput way of asking what circuits are involved. And indeed, from the Simons Initiative for the Developing Brain, from the money invested in that, we've just bought our first light sheet microscope, and Matt Nolan and Natalie Rochefort and Ian DeGuid are all we awarded them a grant to get this up and running. And this will fundamentally change the way that we can address circuit activity in these models. And just to give you an idea from the papers that were published in the last couple of years, this is some of the information you can get out. So I just want you to focus on this region in this animal, and this and it's the same region in another animal. The difference, this is, again, the somatosensory cortex. And what they've done, just as proof of concept, is in this case, the animals have their whiskers intact. And in this case, they shaved off those whiskers. And you can clearly see the somatosensory cortex becomes highly active when there's whiskers present and is not active when the whiskers aren't present. And amazingly, what they found, labs around the world have been studying whisker-based behaviors to understand how the brain processes sensory information. In this paper with this new technique, they identified 2 new areas in the agranular insular cortex that we had no idea played a role in sensory processing from the whiskers. And this is just a very good example of why this technique, how powerful this technique is. And we'll be getting it up and running, hopefully, over the next couple of years. Okay, so that's the imaging. What about the models? Well, again, as I alluded to earlier on, the best models for human disorders are actually the human. Right? If we really want to understand what's going on, and we want to make sure that what we're finding in our animal models, we need to be able to go into human models. And this was solved in 2006 by Yamanaka, who went on to win the Nobel Prize for this. I'm sure most of you have heard of induced pluripotential stem cells, but basically what this allowed was to take human skin or human blood cells and transform them with only 4, in the case of Yamanaka factors, 4 factors that regulate the expression of genes. So they're called transcription factors. And using these 4 factors, he was able to reprogram these cells to be able to become stem cells that could then be reprogrammed to become human neurons. Okay? And you can take these from healthy patients or healthy people. You can take them from people with whatever human condition you want. In the case of what we're interested in, it's in the autisms. And you can start addressing the physiological properties of these human neurons in a dish. And these can become very important for platform testing of drugs. So I'll just give you one bit of data from this work that we're doing. This is all in collaboration with Siddharthan and David Wiley and Giles Hardigan and Shona Chatterjee. And this actually, the data I'm showing you, was produced by Shreya Desharma, who's in the Center for Brain Development and Repair in India. So if we just look at the activity patterns of a typical individual of these cells in a dish, you can see these big action potentials, these bursts of action potentials that happen, followed by silence and then another burst. In the Fragile X neurons, you get many more of these bursts, but fewer and fewer action potentials associated with them. And back to the analogy of the ion channels, if we block ion channels much as we did in our mouse models, if we do that in the control tissue, we can actually turn the neurons from typical individuals into looking exactly like the Fragile X cells. So again, back to our hypothesis that there's too few of these channels in the membrane that are regulating the excitability of the neurons. And if we're right, that if we apply the same potassium blocker to the Fragile X cells, then it should have no effect. And that's exactly what we found. So using these human-based systems, we're able to see that what we're seeing in terms of basic neuronal function in our animal models seems to be very, very similar to what's going on in the human, giving us a lot of confidence that what we're doing actually does translate through. So really what we want to do, we can understand the basic biology, but the key things that we want to be able to do are to prevent and potentially reverse these disorders. So in order to do that, I'm going to give another example of of models that we've been developing to directly address the prevention aspect of this. And again, using new genetic techniques, we can make these models much more easily now. And this is, I'm sure many of you have heard of CRISPR technologies that are coming out. Using CRISPR, we can generate, we're not restricted to the mouse anymore. And what we've moved heavily towards in the Patrick Wild Center, but in collaboration with the Center for Brain Developmental and Repair in India, was we invested heavily in generating rat models for these disorders. And indeed, with the Simons Initiative and with Safari, we're generating more, and we hope to have about 20 of these different models for different monogenic forms of autism within the year. So why would we move to rats? Right? What do rats give us that we can't get from the mouse? And even though mice and rats look very similar, just the rats are a bit bigger, they're actually very different in evolutionary terms. So rats and mice, their last common ancestor was 12 million years ago. And to put that into perspective, the difference between humans and chimpanzees is 6 million years. So these are mice and rats are very different species. And what it allows us to do is now we can, whatever we find in the mouse, or now in the rat, we can always go to a very different species and see, does it cross to another species? And that gives us, again, more confidence for being able to say this is likely to translate through to the human condition. But the other thing that rats give us, because of their behavior, is their cognitive flexibility is far greater than the mice, and we can do much more behavioral assays. There's a whole range of reasons why rats have some advantages over mice. Now, they won't overtake mice. We need them side by side. We need more models, not fewer. But for all of these reasons, we move to the rat. I'm going to show you some data now on whether we can prevent the emergence of phenotypes associated with the loss of FMR1 in a rat model of Fragile X. And we can start asking questions, not simply about the seizures, but we can, or some of the irritability problems, but we can start asking about associative memory and more cognitive tasks. And I don't have time to go through all of these tasks for you now, but they're all based on an animal's desire to explore what's novel. And by changing the environment, by changing the object, by changing the position of the object, we can start tapping into different regions of the brain, because each of these different tasks requires different circuits in the brain. Overlapping, but different. And when we did this with the FMR1 mutants, we found that in all of the wild-type animals can do all of these different configurations of these tasks and identify what's novel. The FMR1 mutants could do some of them, but they were unable to do what we call OPC, or object-place context, what they had to bind together the object, its location, and the context in which it was previously seen. And armed with this now, and having a rat model, and because these are spontaneous exploration tasks, the animals just want to explore what's novel, by just changing up the objects, we can actually test them repeatedly, and we can test them across development, and start obtaining developmental trajectories for when typical animals can acquire this, and whether we can see an alteration in the developmental trajectory of the FMR1 models. So you can see that these are all in weeks down here. So this is about a month of age, about 2 months of age, and so on. And you can see for the easiest of these tasks, the object recognition, wild-type animals can do it from the earliest times that we test them. Similarly with the object context configuration. For object place, this is a bit more difficult for the animals, and they don't actually acquire the ability to do it until closer to the end of the second month. And for object place context, something similar. They can acquire it late. When we look at the FMR1 models, or the Fragile X models, you can see that they're pretty good at doing the object recognition and the object context, and from the earliest time that we've tested them, just like the wild-type animals, they can do it. In contrast, for object place, this is delayed. So where the wild types acquire the ability to do this task at about 2 months of age, the knockouts don't, or the models of Fragile X don't acquire it until several weeks later. And as I showed you before, for this configuration of the tasks, they can never do it. So even in the adult animals, they can't do this configuration of the task. So armed with this, we can start now doing essentially preclinical drug trials in these models and seeing whether we have drugs at our disposal that can actually reverse this developmental trajectory or prevent, in this case, this developmental trajectory. And the first thing that we did, we looked around for what drug we should try. And Emily Osterweil and Mark Baer came up with a very good reason, a paper that said lovastatin, this drug is classically used for high blood pressure. If you look at the biochemical pathways, there's very good reason that lovastatin might be able to have an effect in Fragile X. And they actually published a paper showing that it could help with the excitability deficits and the seizures associated with this disorder. So we were very curious to see whether this could rescue the cognitive deficits. And all this work was done by Antonis Asiminis, who is a joint PhD student with myself and Emma Wood, who's another great colleague here that is in the audience. And here's what I've shown you before. At 7 to 9 weeks, the animals cannot do object place, the FMO models, and they can't do object place context. If we feed them lovastatin in their diet, object place was completely rescued. So now you cannot distinguish. And we fed them the lovastatin in their diet starting at 4 weeks of age, just as this ability to do object place is coming online in wild-type animals. What about the object place context? Well, again, we were able to completely rescue it. So at this point, I ran to Antonis and said, take them off the lovastatin, right? Remove that from their diet, because we need to know whether this is a permanent effect or whether once you take them off the drug, they'll just go back to not being able to do the tasks. And because object place, if you remember, it was just a delay, 4 weeks later off the drug, the knockouts would be expected to be able to do it anyway because it's just a delay. And indeed, that's what we found. But it was critical for object place context where this deficit is found out into adults. And to our amazement, if we test them at 15 weeks of age out here, they were still able to do it, despite being off the drug now for 5 weeks. So we let them grow up longer and change the objects. And even at 6 months of age, these animals are still able to do the task. So this brief early treatment, when normally these phenotypes are emerging or these cognitive deficits are emerging in the Fragile X, if we rescue them at that point, we can then remove the drug and still have permanent rescue or long-lasting rescue. Okay, what about reversibility? So I can't tell you about the Fragile X and the lovastatin because those experiments are still going on in the lab by Antonis. So he's now giving the lovastatin late after the emergence and seeing whether we can reverse it. But Adrian Bird here, who Siddhartha mentioned, who's also the director of the Simons Initiative for the Developing Brain with me, had a great idea. And this highlights that technology can help us a huge amount, but we still need really good human brains coming up with great ideas. And Adrian came up with an idea in Rett syndrome model. He said, can we reverse it if we just re-express the gene? And using clever genetic techniques, he was able to devise a mouse to do this. Now at the time, everybody was convinced these were neurodevelopmental disorders and that there were critical periods that you would have to treat or you would have to do genetic re-expression. And everybody said, Adrian, this won't work. But Adrian said, well, it might. And that's why he's Sir Adrian Bird. But the experiment he did, I'm just going to highlight here. So this is a mouse that's lacking MECP2. So it's a model for Rett syndrome. And these animals get very sick at about 10 to 12 weeks of age, or 6 to 7 weeks of age. They start getting sick, and eventually, if you let them go, they will all die. Okay? So, and this is an animal, just to give you some idea of what these animals look like. Here's a video of this animal. And you can see that it is rolling. This animal is very obese. You can see it's having breathing problems. So you can see these breathing apneas where it stops breathing. And if left to its own devices, this animal would die very quickly, but it would be humanely killed in most cases. And so and now they're just going to lift up by the tail, and if you look at the hind limbs of this animal, they clasp. And this is classic of a neurodegenerative disease, is this hind limb clasping. If we look at this animal, and believe me, what video I'm going to show you is of the same animal, but what I've lost my video. This is just over a month later, and this is the same animal having had MECP2 turned back on. And you can see it's lost the weight, it's able to run around the cage, its breathing has been restored. Stuart Cobb, who's in the audience, I believe, did some physiological recordings, and brain plasticity was returned back to normal. And in a moment, they're going to lift the animal back up by its tail, and you'll see that there's no more of this hind limb clasping. The animal splays its legs as a typical animal would. And if you just look, obviously, if you look at the survival now, that these animals survive well out into adulthood. This was astounding, because what it told us was that what we thought was a neurodevelopmental disorder that would have a critical period for when you'd be able to treat, even treating into what would be considered a young adult in a mouse, completely restored function. Although it wasn't quite complete, there was still some motor gates, but that's pretty good. Right? And it really changed the way we think about neurodevelopmental disorders, and now we're trying to stratify them based on are they true neurodevelopmental disorders with critical periods, or should we be thinking of them like Rett syndrome, where it might be a problem of neural maintenance. And it doesn't matter when we treat, as long as we can get the neurons functioning well again, they'll be able to subserve good function. So the obvious thing that we want to do is, with all of this beautiful animal work, we still need to. The end goal is to get to clinical trials. And as I mentioned, there's reason for hope. There's about a dozen clinical trials going on now for Fragile X, for example. But we shouldn't necessarily expect too much from these clinical trials. Most clinical trials fail, right? Most clinical trials don't work for a variety of reasons, not just because the drug doesn't work. We've been through this, and Andy Stanfield and Andrew McKechnie here were part of the first big multinational trial in Fragile X syndrome that were run by Novartis and Roche. And although these trials were unsuccessful, I would not say in any way, shape, or form they were a failure. They led to many more clinical trials, and they taught us a lot. So they taught us that we have to do better with our preclinical models and identifying core phenotypes, and we're doing that with the imaging and with our clever genetic tools and developing new models. They told us also that we need to be much better once we have potential therapeutic interventions, that we must have to do much better with the trial design, and it's led to big changes now in how we can conduct clinical trials. And it also broke down barriers. So in the first of those two trials, Andy and the Patrick Wild Center were the only site doing those clinical trials. 2 years later, because Andy was able to break down these barriers, there were 6 different sites involved in the Roche trials. And this just highlights how important it is at all times that you have centers where the basic researchers and fundamental researchers are talking to the clinicians. And that's the incredibly rich environment we have in Edinburgh to try to take us through this potential for molecular medicine, this virtuous circle. And ultimately what we're trying to do is to be able to show graphs like this for the neurological disorders. So just to finish up, I have a few thank yous to say. First, for the amazing families that we work with as much as we can, and especially in this talk, for sharing these videos of Ava, Chelsea, and Ken's to really hope you get an idea of the types of disorders we're studying. We have a big thank you to the Wild family, after which this center, the Patrick Wild Center, was named. We finally, just about 6 months ago, this was our first glimpse of Patrick Wild, when Dr. Wild's niece sent us this as she was going through his things. And this is Patrick Wild. We believe he was about 7 years old. And you can see he lived from 1910 to 1955, when there were very few autism services. And this is Alfred Wild, who left a legacy pledge for us to initiate the Patrick Wild Center. And it was in memory of his brother Patrick, and an honor to his parents, who had a very difficult time living and dealing with Patrick throughout that. It was just an honor to how much commitment they had to the family. Again, at a time when there were very little services and very little understanding of these complex conditions. This is at the opening. Here's Timothy O'Shea, the principal, with Alfred. Unfortunately, Alfred passed away just over a year ago. But he's been a great benefactor to us and just a wonderful person. And this is his family, his 2 sisters and his niece. And of course, many things went into setting up the Patrick Wild Center. Here's Andy, who's the clinical director of the Patrick Wild Center. Eve, who Samantha mentioned, who was instrumental, and Chloe Kippen from Developmental alumni, who none of this would have happened without all of these people. And this is at the launch. I need to say a big thanks to Gus and Reem, the parents of Ken's, who have been with us at every step along the journey and do the most crazy fundraising. The first one was they climbed Mount Kilimanjaro. Then they drove from London across the Sahara to Timbuktu in cars that, for some reason, Gus said they're not allowed to cost more than £1,000. And most recently, they've driven from London down the Silk Road to China, all to raise awareness and to raise money for the Patrick Wild Center. And finally, to the Center for Brain Development and Repair that Sir Arthur and I initiated with David Wiley and Chrono Chatterjee, who's based down here. We're on the top floor down here. And unlike when I show pictures of Edinburgh bathed in sunshine, Bangalore is always bathed in sunshine. And it's a wonderful place, and you're all welcome to come visit. And that's the team, the big team down there. And again, even Siddhartha and I never would have been associate directors, but we never would have been able to do it without a total buy-in from all our Edinburgh colleagues. And just to highlight some, there's Shona Chatterjee, who's the director of the center, who's located in Bangalore. There's Ollie Hart, Emma Wood, Mike Cousin, David Wiley is in there somewhere, I can't remember where he is, and there's Siddhartha over there. And it's with the buy-in and the support of all of our colleagues that we're able to do these things. And most recent, it's just started less than a year ago, with, as Siddhartha mentioned, the large grant from the Simons Foundation Autism Research Initiative. We've just launched the Simons Initiative for the Developing Brain. And here are all of our wonderful colleagues, many of whom are in the audience, that will hopefully help us to make some amazing discoveries over the coming years. And finally, you might have noticed that I threw in lots of very, very pretty pictures throughout this. And one of the big things we always do is public outreach. And all of those pictures came from a wonderful collaboration that we have with Sophie Dow, who's here, who is the director of the Mind Room. And we put together all of the wonderful things that we see down on microscopes, and had a huge open-air exhibit several years ago now in St. Andrew's Square. So many thanks to Sophie for getting Sally and I, and Sally did most of the work, Sally and I off out of the labs and bringing a lot of these beautiful pictures to the community. So on that note, so thank you very much.